state next calls Pamela Taylor.
Ms. Taylor, you can come up to the witness stand here. When you get up to the chair, please remain standing and raise your right hand to be sworn in by my clerk.
Do you swear that the testimony you're about to give in this matter before the court shall be the truth, the whole truth, and nothing but the truth, so help you God?
Yes.
Thank you. You can have a seat. When you are situated and close to the microphone, please start by stating your name, spelling your first and last for the record.
Pamela Taylor, Pamela Taylor.
Ms. Taylor, how are you employed?
I'm employed with the Wisconsin State Crime Laboratory in Milwaukee as a DNA analyst.
How long have you been with the Wisconsin State Crime Lab?
Over 12 years.
Have you been a DNA analyst that entire time, or have you worked in any other disciplines?
I primarily work in DNA for about 7 years of that time. I also cross-trained into crime scene photography. So I did that with a crime scene team and then resigned from that about 1.5 years ago.
What are your duties as a DNA analyst with the State Crime Lab?
My primary duties include taking items of evidence, examining those items to possibly develop DNA profiles, and then doing the same for any standards or known profiles, and then comparing to either include or exclude people from the evidence.
Is the State Crime Lab in which you work at accredited?
Yes, it is.
What does it mean to be accredited?
Accreditation is an outside agency or laboratory that comes into our laboratory to verify that our protocols and the training and the process that we do for DNA analysis is accurate and consistent with the forensic community.
Madam Clerk, can you please place the screen on just for the witness? Ms. Taylor, I'm showing you what has been marked as State's Exhibit number 111. It's a 4-page document, and as I'm scrolling through it at the end, I'll ask you if you recognize what this is.
Yes.
What is this?
This is my statement of qualifications. This is basically the training, the number of times I've testified, and any presentations that I've given and training I've received over the last 12 years.
Do you update this throughout your career?
Yes.
And is this the most up-to-date one you have as of today's date?
The only thing that has changed on this since this was submitted was that I testified yesterday in a different court case, so the amount of testimonies would be a little bit higher.
So you've been very busy this week, is that fair to say?
I'm very busy every week.
Okay. Now, can you please explain to the jury your education?
I hold a Bachelor of Science degree in molecular biology, as well as a minor in chemistry and a minor in forensic science. I then completed a training program at the crime laboratory, which included the extraction process of DNA and the instrumentation we use to develop DNA profiles, interpretation of those profiles, comparisons, report writing, and technical review.
As part of your education then, do you also have continuing education in the field even once you become a DNA analyst?
Yes, each analyst is required to do a minimum of 8 hours continued education per year, as well as reading 4 literature articles and proficiency testing, which are 2 tests that are submitted to each analyst. And these are basically run as a mock test or mock trial, as if we were working actual casework, and then we submit those results back to the agency that we get those from, and those have to be accurate and consistent with the results that we were supposed to get.
Now, when I'm looking at pages 2 and 3 of your statement of qualifications, these are 2 full pages where you list approximately 70 items, or I'm sorry, 70 other trainings or continuing educational courses that you have attended over your career. Is that fair to say?
Yes, that's correct.
Have you always been in your career a DNA analyst with the Wisconsin State Crime Lab, or do you have any other scientific employment prior to coming to the crime lab?
I worked as a microbiologist 2 years prior to working at the crime laboratory in a food processing facility, and then transferred over to the crime lab and have been doing this ever since.
Okay. Now, are you published or have you given any presentations regarding DNA analysis?
I have not actual published articles, but I have written articles and done presentations on DNA analysis for different school projects, colleges, and then I also did mock crime scene training for the University of Whitewater.
Does it say on here approximately how many times you have testified at trial about DNA testing and identification, in addition to what you just talked about happened yesterday?
Yes, we keep track of the amount of times we've testified, and including this one and one yesterday, I have testified 164 times in court.
Have you ever been asked to keep track of or been asked the question of approximately how many items of evidence you have looked at in your career for DNA analysis and identification?
I have been asked to approximate the number. Each case is different as far as how many items are submitted and the complexity of them. 1 year, I did keep track of an approximate number, and it was 110,000 samples that year.
110,000 samples in 1 year.
Yes.
When we talk about how those samples are received to the crime lab, can you describe for the jury what the process is once a law enforcement agency either drops off items for testing, or are they sometimes mailed in for testing?
Yes, once the laboratory receives items of evidence from different crime scenes or investigations, it is then given a specific case number, and that case number is specific to either the Milwaukee, Madison, or Wausau labs, and then also given the year and the number case that has been submitted. Then each of the items are given a item designation, either number or letter, depending on what year it was, and then those items are then stored in a secure storage area until the analyst is ready to work them. We then take those items into custody. We work each of the items of evidence first, and then extract those samples, run them through the process to possibly develop DNA profiles. And then we do the same afterwards with standards or known profiles, and then we can make our comparisons.
Okay. Now, when we talk about a report number being created for these items or this particular case, did it used to be designated as an R number, so maybe R241234?
Yes, R was the representing number for Milwaukee, because Madison is M. So yes, we used R, and then 24 would be the year that it was submitted, 1994. And then the year, each item, or each case that is submitted, so if it's case number 1000, that was the 1000th case that has been submitted at that year.
Now, as part of that intake process, are the items received for testing always checked to ensure that there is no tampering or broken seals or anything that would affect the integrity of that sample?
Yes, the evidence specialists look over each of the items of packaging to verify there's no holes, any tape missing, and that the initials and date of the person that had sealed that item are present on that packaging.
If the evidence specialist noted any kind of broken seal, missing sticker, signatures, etc., if they noticed an issue like that, would that have been notated somewhere on or on paperwork related to that sample?
Yes, it is. There is a case file for each case that is submitted, and any issues like that would be noted in the case file.
Okay. Now, let's say when you receive an item and it receives a designation from the crime lab as item A, are there times where an item may be broken up and given sub-designation numbers. So let's say A1, A2, A3.
Yes.
And are those sub-designations always listed separately on any report that you or any other analyst in your lab authors to ensure that we can track which item is being tested?
Yes, each item of evidence, if it is split into multiple items of evidence, will be given a specific designation number. So if I'm working item 1, then even in another discipline, they're going to know what item 1 is, and any sub-designations from that will be referred to in my notes.
And have you seen it in your experience, could one item be submitted to the crime lab and law enforcement request various disciplines test that item?
Yes.
For example, if someone submitted a firearm to the crime lab, the law enforcement department could request a firearms identification or DNA analysis or latent print analysis.
Yes.
Based upon the nature of the type of analysis being requested, would certain disciplines test the item first before going to other disciplines?
Yes, it will.
Okay, can you give me an example of, let's say, a firearm? Let's say the department wanted both prints and DNA and firearm identification.
If we receive a firearm at the crime laboratory, DNA will typically swab that item first. This is basically because we are trained not to cross-contaminate by wearing gloves and making sure that our DNA does not get on anything. The firearms examiners typically just analyze the firearm and the cartridges or the fired evidence. So they are not as trained to handle it without gloves. So if I have a firearm, I will analyze it first, do my swabbings. If fingerprints or the ident department also has to work that item, we typically meet together and we will swab for DNA on the rough surfaces of a firearm, and fingerprints will typically leave analysis for the smooth areas because it's easier to get a fingerprint off a smooth area of a firearm. And so we won't cross what we're testing.
Okay. Oh, I'm sorry, go ahead.
No, that's it.
Now, once or in between testing, is that sample kept, I think you said, in a secure area of the crime lab?
When an item is first submitted to the lab, once it goes through the labeling designation, then it is put into a secure area. So a firearm will be stored in the vault in the firearms department until it's ready to be worked, in which case I would go retrieve it from the vault. If it is a DNA item that it does not require firearms, it is typically stored in a walk-in freezer. And then once I receive that item, I have a stocked storage area that I keep my items of evidence and then return it to wherever I need to for another analyst to lock it into their secure area.
Once all requested analyses are done on a particular item, is the item then repackaged and resealed and relabeled once it's finished?
If I have an item such as a firearm and I have sub-designated that into, say, item 1A, B, C, and D, I will designate that on the outside of the box. I will write that out on the outside of the box or add additional labeling. That way another discipline will know all of those items are contained within. And then, I'm sorry, what was the second part of your question?
And then once we know what items are contained within, is it then sealed with tape or initials or date or anything like that?
Yeah, so every time an item of evidence moves from one discipline to another, it needs to be resealed and then signed and dated by that analyst, and then it goes to the next discipline.
And then once an item is completely finished with any testing from all the disciplines requested, does the item then get sent back to the police departments?
Yes.
Do they usually come and pick up those items, or are they mailed back, or something else?
It could be either one.
Okay. Now, shifting gears completely, a little bit to DNA. What is DNA?
DNA is the biological material found in all living things. Each person will inherit half of their DNA from their mother and half from their father. Each person's DNA profile will be unique to that individual, except in the case of identical twins. It is the same throughout your body. It won't change over time. And it is found in every cell of your body except for red blood cells because they lack the nucleus.
If I touch something or handle an item, does that automatically mean my DNA is definitely going to be on that item?
It's possible that either the DNA is too low to recover a usable profile, or it's possible that the DNA was not transferred.
Okay, so if we're talking about the transfer of DNA via touch, would that be skin cell transfer?
Yes.
And are there a number of factors that can affect, first of all, how much DNA is deposited and whether or not there's enough for you to detect in your analysis?
Yes.
What if, or could the surface of the item you're touching affect that?
Yes.
So for example, if I touch my cup here that has a smooth surface, because it's smooth, is that in your training experience less likely to hold on to DNA?
Yes, we typically, I have found through the different amount of testing that I've done, have found that rough surfaces, like the grip of a firearm typically has a rough surface, will scrape off or retain more DNA skin cells than a smooth surface. So there is no test to determine how much DNA is going to be left in an item based on that, but just in experience, we tend to recover more DNA from an area that's got a rougher surface or has been held longer.
And when we talk about how long you touch an item, that can affect it, or what happens to the item in between when you touch it and the DNA is attempted to be swabbed from that item. Is that fair to say?
That's correct.
Are there also environmental factors that can affect how much DNA you can obtain from an item?
Yes.
What about, when we talk about skin cells, are there also bodily fluids that contain DNA?
Yes, all bodily fluids will contain DNA in a sense. Blood, semen, and saliva are the typical ones that we test for, and we have either confirmatory or presumptive testing. Saliva itself does not contain DNA. It just carries the skin cells from the inside of the cheek into the saliva, and then that gets deposited onto an item.
Okay. Now, when you receive swabs of an item from the police department, let's say, have you seen it where the officers don't send the physical item and instead they send swabs they took of the item?
Yes, that's very common.
Okay. So once you analyze that swab, then you can't look at the DNA and say whether it's fresh or old. Is that correct?
No, DNA analysis determines the DNA profiles that are present on an item if they've been recovered. We cannot tell how it got there or how long it's been there.
Okay. You can just tell what's there and come to a conclusion about the presence of a DNA profile in that sample.
Yes.
Okay. Now, have there been times in your career where you obtain swabs from a police department, or maybe you get the items and you do your own swabs, and through your analysis, there might be not enough DNA information in that sample to come to a conclusion or go on for further testing?
Yes.
And when we're talking about that low-level DNA that's insufficient for testing, does your crime lab have a threshold for the amount of DNA that needs to be present so that it's capable of going on for further testing?
Yes, after the extraction process, which takes the DNA out of the cell material and away from any of the swabs that are submitted, this isolates the DNA in a liquid. And then we do a process to determine the concentration of that DNA, and it has to be over 0.10067 nanograms per microliter. And this is a very small amount of DNA, but it has to be over that threshold, otherwise we know it won't produce a profile. So it's a test that we do to determine how much DNA is in there, and if it's not sufficient, then that sample will stop at that point.
Once you're finished with your analysis on whether it's 1 item or 7 items, do you then prepare a report based upon your findings and your conclusions?
Yes.
Did you prepare a report for the case that you're here to testify about today?
Yes, I did.
So before I start going into that report with you, I had a question about a few phrases that we'll see on this report as we go through it together. When I say single source profile, what is that?
A single source profile is a set of numbers that refer to repeat segments on the DNA chromosomes. And we look at 23 locations, and this will be unique to the individual. So each person here even will have their own set of numbers at those 23 locations. And a single source profile is basically a one-to-one comparison with any standards or known profiles.
Are there times where you are analyzing an item and you see or report a mixture?
Yes.
What does that mean?
A mixture is basically opposite of a single source profile, which is one person contributing, is multiple people. And we can analyze up to 5 people contributing to the profile. So instead of 2 numbers at each of those locations that we look at, if you have a 2-person mixture, you can have 4 numbers and so on. So it makes the analysis more complex and the statistics a little less definitive as if a person is in the profile or not.
Okay. Is it common to see mixtures when you're analyzing DNA on an item?
It's more common to see mixture profiles with touch DNA than it would be for any bodily fluid.
Okay. Now, if you have a mixture, are you able to say which contributor last left their DNA on this item?
No.
Okay. Is it possible to have mixtures that are too low level?
Yes.
And is that for the same reasons we discussed earlier?
Yes, even if the sample gets through the initial process of having enough DNA in the concentration, it is possible that it produces a profile that cannot be used for interpretation based on the limited amount of DNA that's still present. So while it produces a profile, it doesn't meet the requirements that we have at the lab for inclusion or exclusion. So we state it on the report, but we don't use it for any analysis. Can share numbers, and there typically isn't a separation of contributors, so everybody's contributed around the same level. It's hard to tell which ones belong to which people. So also based on our training and our manuals, we're not allowed to make any conclusions on that, so we don't use those as well.
When you are working with a mixture, when you come to a conclusion after you're done analyzing this, do you report that through the use of a likelihood ratio?
If we have a one-to-one comparison, so a single source profile compared to a standard, this is a different statistic. This is called an RMP, or a random match probability, and this is done through a different program. The likelihood ratio is a statistic we use to explain 2 opposing thoughts. The one thought is this mixture is made up of the person of interest, or the comparison being made, and say an unknown individual in a 2-person mixture, or is this better explained, saying this is two unknown people, saying that the types there, or the numbers there, do not match either of these people, and we don't know who these two people are.
Okay. What is a buccal swab?
A buccal swab is a swabbing of the inside cheek of an individual, and this is simply to collect the skin cells from the inside cheek. And this is used as a known standard or a known profile from an individual.
Fair to say you're not the one taking that buccal swab from a known individual. Is that correct?
I've never taken a buccal swab from an individual for
Okay,
a
so
case.
I'm sorry.
No, we've done it for training, but not for an actual case.
Okay, so if you were to receive a buccal swab at your crime lab, that's coming from the law enforcement officers who took that buccal swab from an individual's mouth.
That's correct.
Are you familiar with an STR profile?
Yes.
What is an STR profile in terms of DNA analysis?
STR, or short tandem repeats, are the segments of DNA that we analyze. So a short tandem repeat itself is a segment of DNA that repeats the code over and over and over. So at one location, it may have 17 times that same code repeats, and then that's the number that we use in the location that we use for comparisons.
Sorry, just writing a note. Now, when we talk about using those locations, are those locations what make up a person's DNA profile?
In the type of testing that we do, yes. So we look at 23 locations, and each of those locations will have two numbers. So the collection of those numbers in that order is what makes the DNA profile unique to that individual, and each person's numbers in that order will be different across the world.
Okay. Now, if you are analyzing a mixture, do you use a software called STR Mix or Star Mix?
Yes.
And what does that software do to assist you in analyzing the mixture of DNA profiles that you're observing?
StarMix is a mathematical and biological modeling system. It's a computer program that basically, if I enter in the fact that this is a three-person mixture, saying 3 people are contributing to this profile, it will then separate the mixture into the percentage of each contributor. So one person may be contributing 75% of that DNA. The second person may be 20%, and the last person would be 5%. So it can separate the likelihood of those numbers in each person, and then I can use any standards or known profiles to compare to those percentages in those analyzed set of numbers, and it will give me the likelihood ratio. Is this person one of these 3 people based on those numbers matching, or is this person excluded because the numbers don't match?
When we are going through your report, are there words, or I'm sorry, strike that. When we're going through your report, are there numerical values for reporting your conclusions along with verbal or specific words to describe your conclusion?
Yes, we use 2 different numbers. Well, we have a numerical value, which has a statistic attached to it, which you will see. And then there's also what we call a verbal qualifier. And StarMix uses a couple different levels of verbal qualifiers, depending on how strong that statistic is of that person being included or excluded.
Now, Ms. Taylor, I'm showing you what should be on your screen as States Exhibit 112. It's a 12-page document, and as I scroll through it, if you could follow along with me, and I'll have some questions for you at the end. Do you recognize Exhibit 112?
Yes, I do.
What is it?
This is the report I wrote for R242973.
And is this a result of the DNA analysis you conducted on the various items listed in or on page 1?
Yes.
Is that your signature at the top of the report there as well?
Yes.
And what's the date of this report?
February 24th, 2025.
And at the bottom of each page, starting with page 2, do you have your initials at the bottom of the page as well?
Yes.
Is this a fair and accurate record of the report that you submitted at the conclusion of your DNA analysis in this case?
Yes, it is.
Have you made any additions, corrections, or modifications to this report?
No.
At this time, I would move Exhibit 112 into evidence and ask to publish to the jury.
Mr. Lychwick, any objection to Exhibit 112 coming into evidence? There's no objection from the defendant. It is received. Any objection to 112 being published, Mr. Lychwick? The defendant does not acknowledge the court. It may be used with the jury.
Now, before we get started with the actual testing, I wanted to talk a little bit more about the verbal qualifier that you referenced. If we go to page 12 of 12 of your report, is there a chart here in the middle of the page?
Yes, this is the verbal qualifier chart that represents the numerical values that we receive from the Star Mix program when comparisons are made, and the verbal qualifier is to the strength of that inclusion or exclusion.
So if we were to see on a report very strong support for inclusion, is that the highest level that the crime lab can report?
Yes, typically the statistics that we receive can be higher than that number, but we cap it at very strong support for inclusion because the numbers for statistics can get a little high and unrelatable.
Okay, and when we talk about numbers, are we talking about trillions and quadrillions or 5 and 10?
We're talking about quadrillion and higher.
Okay. Now, is it also possible then, I see on here, uninformative or neutral? Do you see that?
Yes.
Is that the type of scenario where we maybe have too complex a mixture or too low-level DNA, or what circumstances would we see that?
Typically, if you have say a 3-person mixture and you have somebody at a 5% contribution to that profile, that basically means there's not a lot of information of who that person is. Some of the numbers may be dropping out, some information just might not be there. So there's not enough to consistently or confidently say somebody is included or not. So we say, yes, we did the comparison. We don't have enough information to be confident in the answer to that. So we use uninformative to be conservative in our answer.
And then when we're talking about exclusion, is it possible to completely exclude someone versus very strong support for exclusion?
Yes, the more information we have in the profile, so the more DNA that's being contributed. So if you have a 75% contributor or even a 20% contributor, that's a lot of information that we can use. And the higher the contributor, the more information we'll receive. And it can go either way for inclusion or exclusion. So the more information I have and I make those comparisons, either if they match or those numbers are consistent, the statistic will go up and it will go from moderate to strong to very strong support. And the same with, it's the same with either inclusion or exclusion.
Okay. Now, having all of that background, going back to page 1 of Exhibit 112, this list of items we see here, those are all designations that the crime lab has used to identify each of these items. Is that correct?
Yes.
And all of these items would have been taken from the police department, which may have their own number associated with each item based on property inventory numbers. Is that correct?
Yes, our letter designation is going to be separate from the agency's numerical, typically, designation for each item.
Okay. Now, if we look at items C through F, those are ropes from around Carlos Maldonado's body. Is that correct?
That's correct.
Do you know who Carlos Maldonado is?
No.
Do you need to know that in order to conduct your DNA analysis here?
No, it's best that I don't.
Okay. Now, item G, pulled scalp hair from Carlos Maldonado. What was the purpose of the scalp hair? Or I guess maybe a better question would be, what can you potentially do with scalp hair as it relates to DNA analysis?
Typically, when we have a decedent, we will get a dried blood standard from an autopsy, and this is the same as a buccal swab standard from an individual that's living, and this is basically to produce a single source known profile for comparisons. In this case, the condition of Mr. Maldonado's blood was not sufficient to do for testing, so a substitute from the scalp to possibly produce a DNA profile.
And in just kind of foreshadowing, were you able to develop a DNA profile of Mr. Maldonado from this scalp hair?
No.
And why was that?
Typically, when somebody is in the condition inhibits or prevents the recovery of DNA profiles. So while it was attempted, no profile was able to be developed based on the amount of bacteria that was present in the sample.
And then if we move to items Z, AA, AB, AC, and AD. Actually, not yet AD, I'm sorry. But those first four, it looks like swabs from various areas of a pair of boots. Is that correct?
Yes.
Okay, and in this case, you did not actually get the boots at the crime lab. This is clearly designated as swabs from the boot.
Correct.
So the police department would have taken those swabs then and submitted the swabs?
Yes.
And when I say swabs, are we talking like Q-tips?
A swabbing from an item is typically what looks like a Q-tip. It's got a long wooden stem like a Q-tip, but it only has the cotton on one side. And these are typically used by agencies and at the crime laboratory to swab certain areas of physical evidence, and then we cut the swabs off of those sticks, and then we use that as our analysis sample.
Okay. Then going to item AD, that appears to be a pistol with towels wrapped around the barrel and rubber bands and a magazine. Did I read that correctly?
Yes.
And then are there sub-designations for that pistol?
Yes.
And why would there have been sub-designations for this pistol?
When I received the item for AD, the pistol with the towels and the rubber bands, it came in as a whole unit. So when I received it, I then took it apart in steps with a photographer, and I took the rubber bands off, put those and sub-designated those towels. So those were sub-designated separately, and then the pistol and the magazine. So each of the items were separated, and then sections of them were given certain designations before testing was conducted.
Now, when we go to items AE, AF, AG, and AH, those are swabs from various locations on the pistol item AD, is that correct?
Yes.
Do you recall, did you do the swabs from the pistol, or would that have been received from the police department?
If they are listed in this way, they were swabs given to me from the police department.
Okay. And then item AJ, is that a buccal swab standard from an individual named Kevin W. Lychwick?
Yes, it is.
Do you know who Kevin Lychwick is?
No.
Do you need to know who he is in order to perform your DNA analysis?
No, I do not.
And just like Mr. Maldonado, it's better that you don't know.
Yes, it's a conflict of interest if I know anybody in any of the cases that I analyze.
Okay. So now, when we go to the screening portion of your report, which is page 2, what are we talking about with this section here?
If any screening testing is done, such as the possible identification or testing of bodily fluids or hairs, things like that, this is summarized in this section of the report.
Okay. So when we talk about the rope from around Carlos Maldonado's ankles, blood was not detected on that. Is that correct?
Correct.
And then when we talk about the interior blue towel around the barrel of the pistol, blood was not detected on that.
Correct.
When we talk about the exterior blue towel on the pistol, was blood identified on that?
Yes.
And if you are saying blood was identified, does that mean it's the presumptive screening, or did you do the confirmation testing to say that this is blood?
There's two different tests for blood. One is a presumptive test called phenolphthalein testing or Kasselmeyer test. And this basically says I have a red-brown stain or the police suspect that there could be blood on this item. So I will take a small piece of that and then test it with two different chemicals. And this is a color indication test. So if it turns like a hot pink color, it means it's positive, but this is still a presumptive test. It says it could be blood. This also cross-reacts with other things, such as metals, copper, things like that. A confirmatory test says, is this blood, is this human blood? And this will only be a positive test if it comes back as a positive result for human blood. It also does cross-react with weird things like regular ferrets and
Now, when we continue going through your report then, any item that came in that received sub-designations, is that what we see here in this chart at the end of page 2 and then going into or onto page 3?
Yes.
And is this then how you can track each item and what's tested and what the results are?
Yes.
Okay. As we go to this DNA analysis section here, once you start processing these items for DNA analysis, are there some items that you attempted DNA isolation, but you were unable to develop profiles from these items?
Yes.
And would those be the items where we see the words inconclusive or present but limited?
Yes.
Okay. And for the present limited, does that mean that there just wasn't enough of a concentration of DNA?
Yes, so when the test is done to determine the concentration, and it has to be 0.0067 nanograms per microliter, if it is below this number, then it is called present limited, meaning, yes, there's DNA present, but it's not going to produce a profile, and it's under that threshold.
So when we look at in the middle here, where my cursor is, we have items AA1, AB1, and AE1. Do you see that?
Yes.
If we specifically look at AB1 and AE1, those are swabs from the magazine of the pistol. Is that correct?
I believe so, yes.
Okay. And when we look at that section here, it says present, and then autosomal STR. Is that correct?
Yes.
Okay, so when we see those answers in each of the columns, does that mean you went on for additional DNA testing then?
Yes, the first present basically means there is enough human DNA, it's over that threshold, and we can continue on with the DNA testing process. There's also part of that test that determines the amount of male compared to human DNA present, which basically isolates just the Y chromosome. And this tells us how much that there is male DNA present, and it actually gives us a number of how much could be in there. And then autosomal STR is the type of testing that's done that creates the DNA profile based on those short tandem repeats or small segments that repeat.
Okay. And now I'm going to be looking at this column here where my cursor is, but I'll go back up in your report so we can find the correct item that I'm referencing. So what I was looking at is AD1A, which is the swabbing of the barrel of the handgun, and AG1, which is swabs from the slide of the Luger pistol. Do you see that?
Yes.
So now if we go back to that line in your report, we can see that present limited and then unsuitable. So as it relates to those two swabs from the pistol, what were you able to get any DNA from those?
Those both had DNA in the samples, but were both insufficient and under that threshold, so they stopped at this point. And unsuitable just means it's unsuitable for further DNA testing.
If we look at AF1, that sample, which is the swabs from the trigger of the Luger pistol, what was the result of the DNA analysis from the swabbing from the trigger?
This was overall, the human DNA was present limited, meaning it was under the threshold, but it had male DNA. So the male DNA threshold is much smaller. It's 0.002 nanograms per microliter. And this, again, targets only the male Y chromosome. And this is typically used in sexual assaults when we have a female swabbing. So say we have a vaginal swab and there's a lot of female DNA there, and then we have a very small amount of male DNA. What we're able to do is isolate and only type the Y chromosome, and it basically wipes out the female in that profile. And this is also an STR, short tandem repeat testing, but it only targets the male chromosome. So that's typically why we have it. We just do this test for all of the samples that we have. So while it is there, it wasn't used for further testing.
So while we had swabs from the slide and the trigger of the pistol, we were unable to continue with DNA analysis. Is that correct?
Yes, typically when you have two males in the same case, Y testing is a little irrelevant.
Okay. Now, if we go to the bottom of page 3 here, we can see buccal swab standard from Kevin W. Lychwick, which was designated as item AJ1. Is that correct?
That's correct.
And this is a single source male profile. Single source profile. Is that correct?
Yes.
So now this is what we're using to compare to any other DNA profile that we see throughout the testing of these items.
The buccal swab standard is used for comparisons to any of the evidence that has been developed.
Thank you for clarifying. Now, if we go forward then to item AA1, which is swabs from the side of the right boot, we can see here you report a partial single source male profile that's consistent with the profile from Kevin Lychwick. Is that correct?
That's correct.
And then you have a bullet point right underneath it that talks about the probability. Can you explain to the jury, maybe in more layperson's terms, what that is actually saying?
It basically means, so the statistic says the probability of randomly selecting the observed profile from a population of unrelated individuals is not more frequent than 1 in 1 quadrillion. In simpler terms, I would have to type or develop a DNA profile for 1 quadrillion people before I would likely see this same set of numbers in a profile again. So being a single source profile, I'm doing a 1-to-1 comparison. This is one set of numbers, one contributor, and then compared to Mr. Lychwick's profile. And the numbers are consistent, so I can't say 100% that it's him because we have not typed everybody in the world, but the statistic is strong enough to say that it is many times over the population of the world that this DNA came from this person.
If we continue on then to the swabs from the bottom of the left boot, there was a low-level partial profile. So what does that mean?
This means of the locations that we looked at, some of the information is missing. That's why it's called partial. And it's low level, meaning it does not meet the qualifications for interpretation. So it was reported but not used because it is, we cannot be confident of somebody being included in that profile or not.
Okay. Now, Madam Clerk, can you turn on the screen just for the witness, please? You mentioned earlier, Ms. Taylor, that when the firearm in this case was received, it was wrapped up in towels. Is that fair to say?
The barrel of the firearm was wrapped up in towels, yes.
Thank you. And now, there was another individual present who was taking photos of the firearm as it was unraveled piece by piece. Is that correct?
That's correct.
Can you look at the screen in front of you, and I'm showing you a packet of photos that's marked as States Exhibits 158 through 163. And as I go through them, if you could follow along, and I'll have some more questions for you at the end. Okay. Do you see all those?
Yes,
Do you recognize those?
yes,
Are these photos of the firearm as it came in from the police department?
yes.
I would move exhibits 158 to 163 into evidence.
Any objection to the admission of those exhibits, Mr. Lychwick?
This is outrageous. I never saw this gun before. This is not my pistol.
That is not an objection, therefore those exhibits will be received.
Thank you. I would also ask to publish to the jury.
Any objection to publication of those exhibits, Mr.
I
Lychwick?
object to all of this.
Your objections noted. Over the objection of the defense, these pictures may be used with the jury.
In looking at exhibit 158, can you describe for me what you observed about this gun when you first opened the box?
I don't understand your question. If you could restate it.
Sure, absolutely. I'll start with, actually, this box. Is that how this firearm came to the crime lab?
Yes. Well, it came sealed.
It came sealed. Okay, thank you. Now, when you started analyzing this firearm, did you note anywhere in the case file that there was any kind of break in the seal or any evidence of tampering or otherwise concerning that would lead you to believe the integrity of this item might be compromised?
No, upon receipt of this item in the front office, it would have been noted in the case file, and I did not, if I had noticed anything that was not normal in the packaging of this item, I would also note it.
Okay. Now, do we see, once you open this box and you see this pistol, it looks like there's a zip tie towards the grip of the firearm. Do you see that?
Yes, typically firearms are cleared to verify that there's no live ammunition in them, and then zip tied through the magazine area and basically through the top of the gun to verify that there's nothing inside of it before
Okay.
we analyze it.
And where my cursor is, top right of Exhibit 158, is that the magazine for this pistol?
It is a magazine. I am not sure 100% myself because I'm not a firearm examiner if it is belonging to this pistol or not. It did come received with it.
Okay, that's fair. Thank you. Now, if we move on to Exhibit 158. I'm sorry, 159. Is this a closer view of Exhibit 158?
Yes, this is the firearm without the magazine.
Okay. Now, 160?
Yes, this was after I removed the outside collection of rubber bands from the first outside towel.
And as you start removing these rubber bands, are you wearing gloves?
Yes,
And do you have intention on analyzing these rubber bands to see if there's any DNA on them?
yes, I did.
What about the towel? Do you attempt DNA analysis on that towel?
Yes,
As you continue unraveling, if we go to Exhibit 161, do we now see more rubber bands wrapped around the interior blue towel around the barrel of this pistol?
yes,
Do you go through the same process as you did for the exterior towel where you remove all the rubber bands and unravel this blue towel?
yes,
Is the removal of the rubber bands depicted in 162?
that's the removal of the majority of the interior rubber band set. And then once that second towel was removed, the rubber bands that are around the end of the barrel there are separated and added to that collection.
Okay. And then Exhibit 163, what is this?
That is the pistol without all of the towels and rubber bands attached.
Okay. Now, when we look at all of these photos, did you also see a yellow sticker at the bottom of each photo?
Yes.
Does that contain that same R242973 number that you referenced earlier?
Yes.
Does it also have number or the crime lab item designation AD?
Yes.
I am going to bring to you what has previously been marked as State's Exhibit 157. If you look on the outside of Exhibit 157, do you see any yellow stickers consistent with what the crime lab would put on this item?
Yes, this one here is the sub-designation for the firearm and everything in it contained in this box.
Okay, and do you see all this red evidence tape everywhere around it?
Yes.
Obviously now this box opens, so the seal has been, or this item has been opened by someone, is that correct?
That's correct.
And that's not how this was received at the crime lab.
No, it's sealed with evidence tape on all of the on all of the edges, as well as the bottom, to verify there's no, because the gun is then secured with these, and it's taped over so nothing can get into any of the holes or the edges of the packaging.
Okay. Now, if I open then Exhibit 157, what do you see in there?
That is the firearm and the magazine without the towels and the rubber bands.
Okay. Now, this, as we talked about previously, has been rendered safe. The magazine is removed from the pistol, and the all of the bullets are on the outside of the box here. Do you see that?
Yes.
Okay. Now, as we continue going, and Judge, I'm just going to walk this in front of the jury before I ask my next question.
This is 157, which has been previously published to the jury.
That is correct.
Okay.
Now, Ms. Taylor, when we were talking earlier about rough and smooth surfaces and which one is a little bit easier or more likely to retain the DNA, the grip of this gun, from what you could tell, was it more of a rough surface or a smooth surface?
Aside from the tape, which produces a smoother surface, the actual grip is more rough.
Okay, and when you received this firearm, as we see in Exhibit 163, that black masking or black duct tape, was that already around the grip of this pistol?
Yes.
Now, if we go back to your report, which is Exhibit 112, but now we are at item AE1, is that correct?
Yes.
And that's the swabs from the grip of that pistol in front of you in Exhibit 157?
Yes.
Were you able to come to a conclusion as it relates to the DNA present on the swab from the grip of the pistol?
This was developed as a single source male profile, meaning there's one contributor to this profile, and at the number of repeats at each of those locations, I have one set of numbers for each of the 23 locations. And then when compared with Mr. Kevin Lychwick's profile, the numbers in his profile were the same as the ones in the evidence profile. So I can say they're consistent. And it gives the same random match probability statistic of 1 and 1 quadrillion profiles in the unrelated population.
So you would have to have profiles from a quadrillion people before you even have a chance of seeing this same set of numbers or this same profile?
Yes.
If we move on then in your report to swabbing of rubber bands around the exterior blue towel on pistol, which is crime lab number AD3A. Were the rubber bands around the interior and exterior blue towel kept separate?
Yes.
Okay. Preventing cross-contamination?
When you have that many items in the possible DNA testing, the items were kept separate to designate one was from the outside towel, one was from the inside towel. But they were also separated into 2 sections each, making 4 different samples, because if you swab, say, all of them together and they all have a lot of DNA, it can prevent accurate analysis because it may pick up too many minor profiles within it.
Okay. So I am bringing to you what has previously been marked as State's Exhibits 142 and 143. Do you see on each paper bag the yellow crime lab sticker?
Yes.
And does that belong to, do these 2 bags then contain evidence from the crime lab that you analyzed?
These each contain, this is the rubber bands from the first exterior towel, and these are the rubber bands from the interior towel.
And do you see AD4 and AD3?
Yes.
Okay.
Attorney Thompson, when you're done handing the witness the exhibits, I'd ask that you continue your questioning at council table unless you need to be up here for a particular purpose.
Okay, thank you. Now, for Exhibits 142, if we look there, what's the crime lab number on that yellow sticker?
AD3,
And then Exhibit 143, what's that number?
AD4.
And when you look at both of these exhibits, are they currently sealed?
Yes.
And by sealed, do you see evidence tape?
Yes.
Okay. I'll take exhibit number 142, please. And actually, as I look on the back of this, do you see a date and initial on the back of that bag?
Yes. Once I reseal each of the items of packaging, I tape them and then I initial and date the day that I sealed them back up.
Okay. So now, knowing that this item was sealed by you, and it still is now, in opening it with a pair of scissors.
Attorney Thompson, why don't you have the witness open that?
Okay.
She can open it, you can return to council table for the questioning.
Okay. I will leave these with you, and I will certainly get you a pair of gloves. If you so choose to use them.
I do.
There you go. And you can go ahead and open exhibits 142 and 143 and let me know when you're finished.
Okay.
Okay. Now, looking inside of those, we see those rubber bands. Is that correct? And I'm specifically talking about item AD3.
Yes.
Now, in this DNA analysis, we see a 3-person mixture. Is that correct?
Yes.
Okay. Now, how then do you, or at this point, we're using that star mix, right? Because we have a mixture of DNA profiles observed here.
Yes.
Okay, so then now, in looking at this chart below, can you explain to the jury what your conclusion was based on the analysis of these rubber bands around the exterior of the towel?
So the exterior rubber band swabs were separated into two samples. So AD is the firearm, and then AD3 are the rubber bands on the exterior of this firearm around the towel. So then there's AD3A and B, and that just is basically those rubber bands separated into two samples, A and B. So this is the first set of rubber bands. They're not different than the other ones. They were just separated into little piles. So in this one, it is a 3-person mixture, so 3 people are contributing to the profile. I can say at least one of these is male. And then this was separated into the percentages of those 3 contributors and then run against 2 different standards. And Mr. Polly is a very strong supporter of exclusion, meaning he's most likely not one of those 3 people. And then Mr. Lychwick's profile was compared, and it is very strong support for inclusion that this profile is made up of Mr. Lychwick and 2 other unknown individuals than if it is made up of 3 unknown people.
That person of interest and 2 unknown people, that's the better explanation for what you're seeing in this DNA analysis. Is that correct? Or a different way to look at it?
If you see the H1 and H2 in the chart, H1 is the proposition or the idea, is this profile best explained saying that it's made up of the person of interest and 2 other people? Or is it better explained saying this is not made up of that person, it is made up of 3 random unknown people?
And we see that as it relates to the likelihood ratio for Mr. Lychwick, we see that 1 quadrillion number again. Is that correct?
Yes.
Continuing through your report, we now get to a swabbing of rubber bands around the exterior towel and the interior towel with different sub-designations. Is that correct?
Yes.
And while you were able to obtain DNA profiles from these particular areas, were you able to either identify a single source profile or a mixture?
In this case, they were both mixture profiles.
Okay, and were you able to come to any conclusions like we just saw in the chart before?
So these AD3B and AD4B are the second set of swabs from the interior and the exterior rubber band sets. And while DNA profiles were developed, we also do a process in StarMix called a database search. And this goes through all of the samples that we submit into StarMix. And there's a database of collection of people within the lab, people that have visited the lab, and it runs all of our profiles to detect any possible contamination or close match to other profiles of somebody within that laboratory. In this case, both of those samples were found to be a close match to a possible other profile, so they weren't used because they weren't confidently not compromised in any way. So while they were reported, they weren't used for any interpretation of inclusion or exclusion.
They were unreliable for reporting purposes. Is that fair to say?
Yes. The database search is a quality control measure that we have within the lab to verify that if a profile possibly matches other people, that it's not used, and that we note that in our reports.
If we continue moving through your report then, we're next looking at the swabbing of rubber bands around the interior blue towel on the pistol. Is that correct?
Yes.
And you've already opened those items on the stand that are item AD4, correct?
Yes.
Okay, now on these swabbings of the rubber bands from the interior blue towel, you identified a 3-person mixture. Is that correct?
Yes.
And now, can you explain your results in analyzing that mixture?
So this is the first set of the 2 samples taken from the interior towel rubber bands. And this was also a 3-person mixture. At least one of them was male. And this has the same proposition. Is this profile best explained that it is made up of Mr. Lychwick's profile and 2 unknown people, or is it better explained that it's 3 unknown people? In this case, it is a very strong support for inclusion that Mr. Lychwick's profile and 2 other people better explain the evidence than if it is made up of 3 other people.
And again, we see that 1 quadrillion number, is that right?
That's correct.
Now, when we move to the exterior blue towel, that's item AD5, is that correct?
Yes.
And then it has sub-designations for various either parts or cuttings from that towel. Is that fair to say?
Yes.
When we look at that, Ms. Taylor, I provided you brown bags that are marked as exhibits 141 and 148. Do you see the exhibit sticker on there?
Yes.
Are the contents of those bags identified by a yellow sticker from your crime lab?
Yes.
Starting with exhibit number 148, which is the exterior blue towel, can you open that? And can you unroll that, please? Now, Ms. Taylor, you're holding up what's marked as State's Exhibit 148, which is the exterior towel. You reference on your report there's a cutting that you analyzed. Did you cut this towel?
Yes.
Where did you cut it?
Here and here.
Okay, so on the side of the towel that your right hand is holding, we can see maybe two small areas that look like holes, and those would be the cuttings that you did to this towel in order to do a DNA analysis on that blood staining.
Yes, one of the samples was confirmatory tested with the human trace test with blood and was confirmed to be positive. The other sample was done with a presumptive test for saliva and was also positive. We have no confirmatory test for saliva, but the sample was still cut for DNA analysis.
And when you analyze both the cutting of the blood staining and then the cut of a stain on the exterior towel, did you observe single source male profiles consistent with each other?
Yes.
Okay, and are they consistent then with the profile from Kevin Lychwick?
Yes, they are.
And again, we see here that 1 quadrillion number, is that right?
That's correct.
Now, item AD5C appears to be another cutting of a stain on the exterior towel, is that correct?
Yes.
In this cutting, we see that there's a 2-person mixture, is that right?
Yes.
Now, can you explain to the jury then your conclusion on AD5C, that particular cutting from that exterior blue towel?
So a 2-person mixture means there are 2 people contributing to that profile. Each of my locations will have 3 or 4 numbers that represent those short tandem repeat sections. And this is, again, separated by star mix into the percentages. So just estimating the percentage of contributors, say if I have an 80% contributor to 20% contributor, this is asking the question, is this profile best explained with Mr. Lychwick's DNA and 1 other person, or is it better explained as 2 unknown people? And in this case, it is a very strong support for inclusion that the profile is better explained with Mr. Lychwick's profile in an unknown individual.
So now, based on what you see here, at 3 different cuttings or locations on this exterior blue towel, we have Mr. Lychwick's DNA profile on.
The probability is higher in those 3 locations, yes.
Okay. Now if we move to actually before I do that, that exterior blue towel that you held up, did you divide it into quadrants as you were analyzing this piece of evidence?
Yes.
And why separate or why swab in quadrants versus just swabbing all the way across the towel back and forth?
In DNA analysis, it's possible to get too much DNA from the sample, and this creates a complex mixture. And as I stated before, complex mixtures cannot be used for interpretation. So in things such as towels and, say, pillows, things like that, where I'm looking for somebody possibly touching it, I don't know where the object came from necessarily. I don't know if the person I'm looking for is specifically on that item. So if they may have touched in like one corner but not the other corner, I separate it in quadrants to make a smaller sample for each of those swabbings to pick up basically less complex mixtures and the majority contributor for each of those locations.
And now, when you analyzed the swabbings from each of those 8 quadrants on the exterior towel, was there, and you can follow along with me in your report, a 3-person mixture observed on each swabbing from each quadrant?
Yes.
And now going backwards to quadrant 1, when we talk about your conclusion and the best explanation for what you're seeing in this mixture, do we see very strong support for inclusion as it relates to Kevin Lychwick as the person of interest?
Yes.
Okay. Now, you should have Exhibit 141 in front of you. Is that correct?
Yes, I do.
Okay. Can you open that, please?
Attorney Thompson, are you moving Exhibit 148 into evidence?
I will be, yes.
Okay, well,
Oh, I'm sorry.
I should have.
I thought I did. If I did not, I would move Exhibit 148 and 141 into evidence.
Mr. Lychwick, any objection to the receipt of Exhibits 148 and 141?
I object to all of this. That's not my pistol. I don't know how my DNA got on there.
Mr. Lychwick, if you want to make a legal objection, you can do that, but editorializing or trying to testify will not be allowed. Do you have a legal objection to the admission of these exhibits? Hearing none, they are admitted.
I object.
Your objection is noted without any basis. These exhibits are entered into evidence.
Thank
You may
you.
continue, Attorney Thompson.
I'm sorry. Thank you, Judge. Now, Exhibit 141, is that identified by the crime lab on that yellow sticker as AD6?
Yes.
And then were there 4 sections that were swabbed on that towel that were sub-designated as AD6A through D?
Yes.
Okay. Now, can you please, if you have not already, can you open Exhibit 141 and hold that up for the jury?
Attorney Thompson, hang on. Are you asking for publication to the jury?
Yes, please.
Okay. Mr. Lychwick, do you object to the publication of this exhibit to the jury?
I strenuously object.
All right. That is noted. The exhibit is in evidence, and it will be published to the jury. So now you may proceed.
Okay, and just so the record can reflect, Ms. Taylor held up the interior blue towel and then she turned it around holding it up in front of the jury. Now, Ms. Taylor, on this interior towel, your report notes swabbings of sections 1 through 4. Is that different from how you analyze the exterior towel in quadrants?
So the exterior towel was swabbed in quadrants 1 through 8, basically. And each side was split into 4 quadrants. So in this towel, it was only split into 2 on each side.
Okay. Now, at the conclusion of your analysis of these 4 sections, did you identify single source male profiles on each of those sections consistent with each other?
Yes.
And are those profiles consistent with the profile of Kevin Lychwick?
Yes, they are.
And when it talks about single source, that's one contributor? It's a one-to-one comparison?
That's correct.
Okay, so in an overview then, when we talk about the exterior blue towel, the interior blue towel, the grip of this pistol, and the rubber bands, whether it's a single source profile or a mixture, we're seeing either profiles consistent with Kevin Lychwick or very strong support for inclusion of Kevin Lychwick.
That's correct.
Sorry, Judge, just one moment. I have no other questions for this witness. Thank you.
Attorney Thompson, I do not have exhibits 142 or 143 in evidence. Are you moving those in?
I am. Yes, please.
Mr. Lychwick, any objection to the admission of 142 and 143?
I strenuously object.
Okay, over the objection, those exhibits will be received. And Attorney Thompson, are you moving exhibit 111 in the statement of qualifications?
Yes. So before Ms. Taylor leaves the stand, I would move exhibits 111, 112, 158 through 163, 157, or I'm sorry, 157's already been admitted, 142, 143, 141, and 148.
I had all of those other ones already in. The only one I didn't were the only ones I didn't were the ones I mentioned. So, Mr. Lychwick, any objection to 111, the statement of qualifications
I
coming in?
strenuously object to this all.
Okay. Over the defense objection, 111 will be received as well. Mr. Lychwick, do you have cross-examination questions?
Yes, I do.
All right, you may begin.
Is it possible for an outside individual to place someone's DNA on an object, to attach them to an object that they had never touched before, perhaps? Is it, in your opinion, that's possible?
It is possible, yes.
At this point, I have no other questions for this witness. Thank you for answering.
Any redirect?
No.
Okay, you may step down. Ladies and gentlemen, it's 10 minutes to noon, so we are going to take our lunch break right now. Let's plan on resuming at 1 p.m. And again, remind you, as I will try to do at every break, please do not discuss this case amongst yourselves yet. You have not heard all the evidence and you have not been instructed. So have a good lunch and we'll see you at 1 o'clock.
All rise.
Thank you. God, thank you very much. Where's my number?
Thank you. You may be seated. Is there anything we need to place on the record before we break, Attorney Thompson?
No, thank you.
Mr. Lychwick?
Yes. Again, I strongly object to these proceedings. I am unprepared to defend myself, and I am sickly. I should be in a hospital.
Mr. Lychwick,
Not
you
prepared
continue.
to be here. And you dragged me here
Please
against
stop
my
talking.
will. I am here under duress, and I protest.
Mr. Lychwick, your objections and your protestations are noted for the record. The court has ruled on those, and the trial will continue at 1 p.m. We'll see you then.